Distinguished speaker, faculties, and students, a very good morning to one and all present here. My name is Marzuka Asad Qureshi, and I'm a fourth year student of Biotechnology Dual Engineering. I'll be your host for today's online expert session. With immense pleasure and with the permission of Honorable Pro Vice Chancellor, Professor Dr. A.W. Santosh Kumar Sir, Director of Amity Institute of Biotechnology, Professor Dr. Aparna Khanna Ma'am, the convener of the session, Dr. Ravi Shankar Patil Sir, and the co-conveners, Dr. Rahul Khandare Sir and Dr. Abhishek Sir. I would like to welcome you all on behalf of the Amity Institute of Biotechnology and Amity University, Mumbai. We are delighted to have you with us to participate and share in our today's event. Thank you so much for coming. The theme of today's online expert talk is Phytoremediation Technology for Textile Industry Wastewater Treatment, a Success Story. This discussion will brush upon a spectrum of exciting and benefiting subtopics that discusses several aspects of our esteemed guest speaker, Professor Sanjay Govindvar's journey of research to develop sustainable phytoremediation technology to clean up direct textile effluent at various industrial sites. He is an eminent faculty member at the Department of Earth Resources and Environmental Engineering at Hanyang University, Seoul, South Korea. Let me recapitulate a few points before we begin with this session. Phytoremediation is a plant-based approach which involves the use of plants to extract and remove elemental pollutants or lower their bioavailability in soil. These have the ability to absorb ionic compounds in the soil even at low concentrations through their root system. Moreover, they absorb, accumulate, and metabolize toxic chemicals and pollutants into non-toxic or less toxic metabolites. Evidence suggests that numerous aquatic and terrestrial plants have the potential to remediate environmental pollutants alone or in combination with microbial species. Our today's guest speaker, Professor Govindvar Sir, has done tremendous research work in the field of phytoremediation as well as bioremediation and has successfully developed plant microbe consortium based technology for toxic textile effluent treatment at industrial sites. Without further ado, I would like to request Dr. Ravi Shankar Patil, Assistant Professor at Amity Institute of Biotechnology and the convener for the session, to introduce the Pro Vice Chancellor of our university, Professor Dr. A.W. Santosh Kumar, to inaugurate the session. We hope you all take everything that you can from this session. Ravi Shankar sir, over to you.
Okay, so good morning, all of you. Thank you, Marzuka. So my voice is audible, Marzuka?
Yes, sir.
Okay, thank you very much. Okay, so it's my immense pleasure to introduce our Honorable Pro Vice Chancellor, Sir Dr. A.W. Santosh Kumar. He has 33 years of academic and administrative experience. He has held the post of Vice Chancellor for a period of 4 years in one of the highly ranked universities. He has spearheaded many accreditation and academic committees, including NAC and NBA. His administrative caliber includes establishment of new institutions and center of excellences in the earlier places where he worked. He has brought in extramural funding to the tune of 1.8 crores. Earlier, he was working in the United States for a period of 16 years and has held various positions such as Dean Academic Affairs, Dean Accreditations, Co-Director Inland Stem Cell Consortium, Co-Director Center for Genomics, Director STEM Program and Professor of Microbiology and Infectious Diseases. Honorable Pro Vice Chancellor Sir completed his PhD from the All India Institute of Medical Sciences, New Delhi, and further completed his postdoctoral fellowship from University of California, National Cancer Center of Singapore, J. Craig Venters Institute, USA, and National Institute of Health, that is NIH, USA. He is currently funded with 5 extramurally funded projects, one from the NIH USA, 3 from DBT, and 1 from DST. His major research focus includes study of human microbiome, role in systematic diseases, which is funded by the NIH USA. His other area of research is on host pathogen interaction and drug discovery, funded by DBT and DST India. His performance indices are H-index 34, I-10 70, and citations 4,717. He has published 181 peer-reviewed research articles, 4 books, 12 book chapters, and he has having 18 patents to his credit. He has guided 15 doctoral candidates and currently guiding 5 candidates. His academic services include sitting members in various extramural funding agencies nationally and internationally, such as NIH Study Section for Grant Review, member DBT Technical Advisory Committee, member International Fellowship.
Ravi,
Yes,
sorry to cut you. I think we can go ahead to the main thing.
okay, sir. So now, without further ado, I request our Pro Vice Chancellor, sir, to deliver opening remarks. Welcome you, sir.
Yeah.
Over to you, sir.
So good morning, everyone. Respected Chief Guest Officer, Dr. Sanjay from Seoul, South Korea. Our most respected Dr. Aparna Kanaji, Dean of Research and Director of Amity Institute of Biotechnology. The organizers, Dr. Ravi Shankar, Dr. Rahul, Dr. Abhishek, all the faculty members, my most revered and most heartful student friends. It's indeed a pleasure that Amity Institute of Biotechnology is actually bringing in new novel knowledge every month with new perpetuations. And this is to cater to the recent trends and to the developments of science and technology with special reference to biotechnology per se. And today, we are going to just listen to a very important lecture, which is on phytoremediation technology, especially for the textile industry, wastewater treatment, and which is very important with regard to the Indian setup is concerned, because the Indian population mainly dwells in good water sources, and these water sources are being contaminated by this textile industry, not only in different pockets of India, but also in a sporadic way. And talking about phytoremediation, though we have different types of phytoremediation, such as rhizofiltration or phytoextraction, phytotransformation, phytostabilization, etc. So overall, the plant-based bioremediation has played a very vital role, and one of the important plants that we use for phytoremediation, as far as I know, is the water hyacinth. So it is indeed a pleasure that we have with us Dr. Sanjay, who's one of the important stalwarts in this area. We look forward to getting a new knowledge from him. And student friends, I should certainly say that getting a person from a different part of the continent talking to Indian people as an Indian, it's indeed a very great pleasure. And I really thank you, Dr. Sanjay Govinda sir, for accepting this and then coming and addressing this. And thank you so much, and I wish you all the best for this one.
Thank you, sir. I would now like to invite Dr. Abhishek Gulde, the co-convener for this session and associate professor at Amity Institute of Biotechnology, to introduce our esteemed guest speaker for this session. We hope that this session inspires ideas and discussions. Abhishek, sir, over to you.
Thank you, Marzuka. Am I audible?
Yes, sir, you're audible.
Okay. So thank you very much. I am elated and feel honored to introduce respected Professor Sanjay Govindwar, who is currently faculty at Department of Earth Resources and Environmental Engineering, Hanyang University, South Korea. Professor Govindwar has research and teaching experience of almost 4 decades. His research focus area is microbial transformation, phytoremediation, environmental restoration and remediation. He has done internationally acclaimed research in the area of bioremediation, phytoremediation of textile effluent. He has developed lab-scale phytoreactors, a wetland, and a phytobed technology for an efficient bioremediation of textile industry effluent and of textile dye contaminated sites. Due to his extensive research work in these areas, he is featured in the list of top 0.1% scholars by Expertscape, World Expert, and top 2% world scientists by PLOS Journal. His achievement also includes numerous international and national awards and fellowships. He is Fellow of Biotechnology Research Society of India, Maharashtra Academy of Science, and of International Society of Biotechnology. He was former professor and head at Department of Biochemistry, Shivaji University, Kolhapur. He has held various positions such as professor and brain pool invited scientist at Hanyang University, Gyeongsang National University, Konkuk University in South Korea, and National Taiwan University, Taipei, Taiwan. He has various prestigious postdoctoral fellowships at University of South Florida, Tuskegee University, USA, Hindustan Antibiotics, and Marathwada University. Professor Govindwar is editor-in-chief for American Journal of Bioengineering and Biotechnology. He has served as member of the editorial board in reputed journals such as Research Journal of Biotechnology, Journal of Microbial World, the Scientific World Journal, Journal of Microbiology and Biotechnology. He has also served as a member of several national and international scientific committees and advisory boards. He has successfully completed several projects in environmental biotechnology area funded by CSIR, UGC, ICAR, DST, DBT, Government of India, as well as National Research Foundation, South Korea. He has worked on projects and technology transfers in association with academia, government bodies, and industry. He was also part of research infrastructure projects such as DBT FIST and UGC SAB TRS. Professor Govindwar has supervised 40 PhD students, 1 MPhil, 2 postdoctoral fellows, and 1 TWAS fellow. These students hold various reputed positions at national and international academic and research institutions. He has attended several national and international conferences, seminars, and workshops and delivered guest lectures. He has 1 patent and 281 research publications. His research has been well received across the globe. His Google Scholar citations are 15,563 with an H index of 67. Indeed, it is our privilege that today we have him here to share his knowledge and wisdom. Hearty welcome, sir. Now I invite you to deliver your expert talk.
Okay. Thank you very much. Now, at the outset, I'm thankful to Vice Chancellor Santosh Kumar of Amity University and Aparna. Sorry, it is very difficult for me to name all the people because everybody is new for me. I'm very much thankful to authorities of Amity University as well, Dr. Ravi Shankar also. I remember him because he's my student. He was working with me since long time.
Yes.
So it was very nice introduction for me. Now, can I share the screen now?
Yes, sir. Yes, sir.
I will start my lecture. I will share the screen and
Yes, sir. It is shared now.
yeah, okay. Thank you very much. Now, this first slide itself is saying something different, that phytoremediation technology for the textile dyes, textile industry wastewater treatment, a success story. Now, the picture what you are observing here is displayed by Department of Biotechnology, I think in 2019 on environmental day. Okay, and they say that green remediation treatment process of textile dyes in wastewater developed. Okay, that is what I say is a success story, and this system is presently working at Kagal MIDC. That is a 5-star MIDC, Kagal, Kolhapur. Okay, that's why I say that is a success story. And it is the important part that how we could get success, because Shivaji University is mostly in a remote area. And we could get success. There are so many reasons, because mostly we were looking for the technology. As well, it was a need-based research, because it was in need. You might be aware of that. Ichalkaranji is nearby city of Kolhapur. There are lots of textile units, and we get a lot of textile wastewater, and that is directly thrown in the rivers. As well, there are some, I can't say that always thrown in the rivers, but reality it is. But in practice, we can say that there are some CETP plants are available, treatment plants are available, but as usual, the plants are not working properly, and you will get the pollution. Okay, and considering this part, we have started our research. Okay, can we go for next slide, please? Ravi Shankar, how you can change the slide? Okay. Now it is a usual pollution scenario. I especially mentioned where the rules and regulation are not followed strictly. It is always, you can find if you go in Ichalkaranj area or wherever the textile industry, small textile industries are there, you will find this type of the things. Not only in the rural area, you can say such an area like Kolhapur, we get a lot of wastewater which is generated having this type of the water, which is always contaminated with the textile dyes. And what are near about, we estimated that near about 15% of the textile dye, what we are used for the processes will be in the wastewater. And we are bothering about that. Okay, and that's why we started our research for the cleaning of the textile wastewater from the industries, what we have got collection from different industries. Now, so water pollution by the textile effluent, why one should go for that? Because the textile is very toxic. They are carcinogenic, mutagenic, allergic, cytotoxic, and genotoxic. And because of that, directly that will be health to the human being. Textile wastewater usually disposes in the river. Mostly what we observe that when it is sample is more, the effluent is more, that cannot be treatable by the established CATP plants, and mostly untreated water is being thrown in the rivers. That is the basic problems. Okay, and what is the current scenario of the available techniques available and the constraint? Mostly in CATP plants, we are using physicochemical methods by which we are producing non-degradable sludge formation, which contains most of the dyes and sludge. Then we have some effluent treatment plants. Truly speaking, that if you use effluent treatment plant very carefully, as per the standard, mostly you can get most of the textile water are getting clean, but somehow it is not being clean. And that's why I say here, partial cleaning of the textile dyes. Okay, but the norms, if you follow the norms with the textile, norms of the effluent treatment plant for the treatment of the textile dyes, yes, you can get it. But not single textile effluent plant is properly working. Okay, and that's why I say the partial cleaning of textile wastewater. Then landfilling, the whatever the non-degradable sludge is formed here, and you can go for the landfilling, and that leachates leading to the water body pollution. Okay, in rainy season, the water is getting absorbed, and again, the whatever the dye is getting accumulated, that will be flown in the rivers. Then pre-discharge, yes, is uncontrolled damage of the environment. As we know that everywhere you can find the colored water in the system. And with this concern is a very serious concern, like water and soil contamination is very serious concern, and one should go for that. That's why we started our research. Next, please. Now, if you're talking about the remediation processes or cleaning of the wastewater, there are some potential bioremediators like bacteria, fungi, and the plants. And these plants and microbes, we can say bioprocessor or biochemical reactions present in this have unlimited power of working together to clean up and the messes produced by the man. Next, please. Now, with this, we started our research as Qureshi has explained what is the phytoremediation very nicely. That is, in short, what she has mentioned. And it is very easy to say like that. But the question first comes in that does plant do this type of the function, or this degradation takes place? Can they degrade the textile effluent? That was the question. And with that, we started with we were working on mostly our lab was working on this microbial degradation of the textile dyes, and everybody was aware of that. Microbes do have some enzymes and they have the capability to degrade the textile dyes. However, phytoremediation of a textile dyes was the new area at that time, and it happened that we were working on the phytotoxicity on Brassica zensea, and what we found that whatever the dye we are added in the control, that is also decolorized by Brassica, and that was a key question for us, the why it is degrading, and we starting the work from Brassica zensea. And first our publication, you can say we have the publication of phytoremediation showing that Brassica zensea is can remediate the textile effluent. And then we started, you know that Brassica zensea cannot be used for the phytoremediation because of it is an edible plant, and these are really objectionable. That's why we started the screening of number of plants for the bioremediation purpose. Question was only that does declaration takes place by using the these plants. So we can see these are the flowering plant we have chosen. And the reason for this is that if you have visited any HRTS, is always dirty. And we wanted that if we can make that HRTS beautiful, that will be better. It's pleasant. And we started with Blumea malcolmi, Aster MLS, Portulaca glandiflora, Glanduria pulchella, and Zinnia west. And we have taken one of the dye here, and we got a different type of the percent variation. This indicate and with different time interval. Now, by this, we could get the indication that the plants can metabolize the textile dyes, and by which we can get the decoloration. And it certainly requires sometimes. And by this, one can understand here also that potential of the different plants are the different one. Next slide, please. Now the question that what we got the plant, and this plant is only uprooted plants, and uprooted plants contains microorganisms, so bacteria, they are coming with the roots. And one of my students raised the question that, sir, okay, because we were working on mostly on the bacteria, and she said that bacteria may be there and bacteria does this work. And naturally, that was the question, that do plants involved in the biotransformation or not? Particularly, I'm talking about the textiles. And here, we work on this plant cell culture, that is Blumea cells we are used here. Then plant callus culture also, and we got the degradation here too. Then we generated the plantlettes, tissue culture plantlettes, and we got this degradation. And then for the confirmation with the enzyme activity, we went for the transgenic hair roots, and from using the transgenic hair roots, we are also got the degradation of these textile dyes. Now at this stage, I will say that decolorization of textile dyes, then we can think of the it is decolorization only or the degradation. Certainly it is a biotransformation reaction. Next, please. Now just to confirm with this, we have cultured tissue culture plants, and we have seen the metabolism of the different textile dyes with different plant isolated in tissue culture, and we got the degradation here in particular times. By this, we have proved that yes, plants are taking part in the decolorization of the textile dyes. Okay, and I think we have published this. These are the whatever the blue line you find here, that is our publication, just to prove that yes, authenticate our research. Next, please. Now only decolorization is not that important. Decolorization is not important because decolorization can be done by change in pH. or just other parameters if takes place, you can get the declaration. But here we are thoroughly studied that now in these figures you can say that we are search for the anatomy of the stain, and we are seeing here because different time interval you can find the dye, and if you keep this cells for this roots cells for 48 hours for tap water, it is also clean here. That means by this we have proven that dyes are entered inside the cell or inside the plant tissue, and they are be coming out. And we got some cell photographs also, what will be the effect of dyes on the unexposed and the exposed cells. These meristematic cells of the roots, and this is showing the decolorization. And you can find your HPLC analysis plate. This is first lane is only dye, and other lanes are the product of this. That means we got the product. Now here you can say the degradation. Okay, so by this plate we can show that there is a degradation of the textile dyes takes place. Next, please. Major part of the degradation, because this research we might have started in 2000, around 2006 or something like that, and that time we could find that lots of research papers were observed on dye declaration. One of my friend also suggested, yeah, there are number of papers present for the this decolorization things, and why you are interested in this area. But what we found that that time, yeah, there were papers, number of papers were there, but nobody talks about the metabolism of the textile dyes, product formation of the textile dyes, and nobody was talking about that why there is a degradation of the textile dyes are taking place. And mostly work was being done in microorganism, and people were thinking that the microorganism require food, and that's why they are degrading. That means they were, if there are deficiency in the food, then they think that yes, there is a degradation of the dye takes place, and that's why they created deficiency of the food deprivation, food deprivation, and they added dye for the declaration. But it is not possible always. And we thought that are the enzymes are responsible for the metabolism. Then we know that on the basis of microorganism as well as in the plants, yes, enzymes are working in this. And we tested with peroxidase, tyrosinase, DCIP reductase, azo reductase, riboflavin reductase. These are the enzymes. Here, for example, we have taken Blumia malcolmi, and this is Portulaca grandiflora. These are the different plants isolated by tissue culture, and we have shown that degradation. And if you see the figures here, 0 hours and 70 hours, there is an increase, a 4 times increase here. That means induction is there. And usually it is being said that if there is an induction of the enzyme, these are the inducible enzymes. If the induction of the enzymes takes place inside the cell, there might be possibility that these enzymes are working for the degradation. Okay, degradation of that particular molecules. Not necessarily it is responsible for that. I'm going to show you just you have to prove, but only that if induction is taking place, yes, that is because of the stress developed by the plant, and these enzymes might be responsible for the metabolism of the molecule which are producing the stress. Okay, this is the simple definition of the you can find in the biochemistry. Now, in general, we are look for these peroxidase, these enzymes, and we have shown that there is an induction of these enzymes are taking place. Probably these enzymes are working for the degradation of the textile dyes. Next, please. Okay, so now you can see the list here. This lignin peroxidase, lactase, volatile oxidase, azoreductase, most of the oxidoreductase enzymes, oxidase enzymes and reductase enzymes, and mostly biotransformations, oxidases are responsible for that, or the reductase are responsible. And here we have seen that is it working in only roots or the stem? Okay, and we found that yes, these are working in case of the in roots also, and as well as you can some enzymes are inducible in stems too. That means degradation can take place in the root as well as in the stem. Okay, next please. Now, as you may be aware of that, the plant cells are very difficult to study. The reason is that there are lots of pigments present in the plant cells, and that pigments is always difficult to they get obstacles in measuring the decolorization, the color measurements, and that's why we have chosen a model of hairy roots and for the purification of the enzymes particularly. And hairy roots are as good as I think most of the that is hairy roots are as good as they are showing all characteristics of the plant. So we use hairy roots. They are very delicate, and you can produce the hairy roots in ample amount. And while you're using the hairy roots, we have seen that all the enzymes like lignin peroxidase, lactase, tyrosinase, amine peroxidase, DCIP reductase, and azo reductase, you'll find that there is enzymes. This after declare when these roots are exposed to the textile dyes, we could find the induction of the enzymes. See all figures. If you compare with these green figures with this intracellular green figures with this intrafigures, this will be induced here. That means we can say here that degradation, yeah, there are enzymes present in the hairy roots. They also take part in the metabolism of this. Okay, next please. Now again, question comes, naturally any biochemist, as I mentioned earlier, any biochemist can say here that induction doesn't mean that it is involved in the metabolism of that particular molecule. So to test that, we have purified plant lackages, and we are using the plant lackages. This is the Blumia lackage from the Blumia hairy roots, and we did this experiment is only you can say inhibition, competitive, non-competitive inhibition, what we have seen in your science, that is absence and presence of the dye, and we got the competitive inhibition. It clearly indicates that yes, Blumia lackage is degrading this dyes. Okay, and similarly, we could find that lackage, lignin peroxidase, azo reductase, they are working for degradation of the textile dyes. Rest of the enzymes, what we DCIP reductase is a common reductase enzymes. You can find induction there, and like tyrosinase is also common enzyme. We should not give the importance for that. And likewise, you can say that by this we could understand that lignin peroxidase, lackase, and azo reductase are really responsible for the metabolism of textile dyes. And we could find here that basic lackage degradation pattern by using GCMS analysis, by identifying the metabolites generated. We could find out the flow chart of the metabolism, how the metabolism pathway is there for the degradation and what is the last product. Surprisingly, we find a difference in the bacterial lackage and the plant lackages, because bacterial lackage doesn't require ABTS as a mediator, but plant lackages requires ABTS is a mediator for the reaction. That may be because of the redox potential of the plant lackages is different than the redox potential of the bacterial lackages, and this is matches bacterial lackages matches for this mostly ABTS or this type of the compounds. So you will be requiring the redox mediator. Next, please. Okay, this another way that you can analyze the sample if you degrade the sample, declarize the sample, then you can go with the HPLC analysis, and you'll find the different pattern of the peaks. You can say this is a control and test like this by using the Ramazal raid you are taken dimixture and treated textile water like this. You'll find the pattern is different. We are not talking about here because degradation of the textile dye is not necessary to go for the exactly what type of the degradation is taking place. Mostly we always think about that we are using the textile effluent. We never know what is present in the textile effluent because the effluent is coming from different industry. Number of dyes will be there. Only that we can see here the pattern. That's why we are not saying that which peak is getting reduced and which peak is getting inserted here. And in the most of the processes also, you can find the generation of some other molecules which can give peaks too. Okay, but only here for HPLC analysis we say the pattern. Pattern is varied, that something is going on. Next please. Now by using these processes, you can get what is the fate of metabolism and what is the product. Okay, and this proposed pathway you can make it with the enzymes which are induced and what is the product form. And because it's a very simple thing, because basic structure, you are aware of the basic structure and you can calculate on the basis of the molecule what is the probable product here. So as you can know, synthesis is something different, but in this case it is very simple to to determine the proposed pathway for the metabolism of that particular compound, particular textile dye. Next. Okay, as I say that enzymes are involved in the degradation. Similarly, I said that it is a degradation, not the only declaration, because it is very important. Declaration doesn't mean that there is a metabolism to the smaller molecule. Okay, and same way you have to see that how long you can use the plants. Okay, so if you are using the plants and within 10 or 40 hours, if the plants get deteriorated or death of the plants, it is of no use for the industrial purpose. Okay, so we have to check what is the health of the plants. And here in this study, we have seen that chlorophyll A, chlorophyll B, and the carotenoids. And suddenly the level is good enough. Just by looking towards the plants, yeah, plants were good. And that's only plants we are selected. But there is an increase in this chlorophyll A and chlorophyll B. That may be because of the generated stress, but stress was not that much that the plant will survive. And such a plants you can use for phytoremediation purpose, like vetiver giganoids. Yes, you can use this plant because there is not that much stress here. Okay, next please. Now, metabolic, whenever we are talking about the biotransformation processes, biotransformation means change of one chemical to another chemical. That is simple biotransformation. But we are not sure that whatever the generated chemical is toxic or non-toxic. Okay, as I mentioned in first reaction that toxic will be converted into non-toxic, but that is not necessarily that toxic will be converted to non-toxic. Okay, biotransformation says one compound is converted to another compound. So it is very important to check the toxicity of that effluent. And we tested this with the plant sources only because mostly this water is being used for agriculture purposes, not for as a drinking water, agriculture purposes. So this agricultural plants we are taking for the checking of the toxicity. Certainly, we have generated some other methodologies also for checking the toxicity of these molecules, but that contest is not here. So I will be only concentrating on these plants. So mostly that when we check this toxicity of the plants, we go to the percent germination, root length and shoot length. And if you find here, percent germination in control dye is 50 and taste is 85, that means germination is increased by by the treatment of the wastewater. This is, you can say, the comparison between untreated textile water and treated textile water. You'll find that treated textile water values are more than untreated textile water. That means toxicity is reduced. That system is working. Okay, and mostly in this remediation processes, we have to bother about the toxicity of the treated textile water. That is very important. You may get the biotransformation, but if the toxicity is more, this is of no use. Okay, so this also we have checked that yes, toxicity is getting decreased. Then we can follow this system. Next, please. Now, as we have seen that the enzymes till now, but if you're talking about the environmental concerns, environmental scientists, they are worried about the COD, BOD, TDS, TSS, electric conductivity, turbidity. They are not worried about the enzymes, what enzyme is induced. Nobody bothers about that in practice. Only, yes, certainly there is a botheration of the color, because this is unpleasant. And here we have tested all these with the vetiver seasonoids, with dimixture and the textile wastewater. And you will find here there is a decrease in ADMI is a color index. There is a change in the color, decrease in the color. COD is also decreased, BOD is also decreased, TDS is also decreased. Mostly if you find in textile wastewater, TDS, it is 4,280, it is decreased to 1,364. This is a very toxic and TDS, that is, this water cannot be used for the agriculture. However, after treatment, this water can be used for the agriculture. Okay. Similarly, you'll find that all parameters also, the parameters, parameters are decreased. Electrical conductivity is increased. That is because of the generation of the metabolites generated there, and that's why you can get, we can find that electricity conductivity is increased. Next, please. Okay. Now, by this basic things, you can say the primarily, which are very important things, that we have to show that yes, plants are taking care of the degradation of the textile. That one part is over. But by using these results, we could observe, yeah, different plants have different characteristics. And that's why we have screened various plants for the degradation of the dyes. Okay, primary, we have started the research on dyes, not the textile wastewater. And certainly we have seen, you might find different different plant and different dye here. And one may say that, okay, sir, why you are not working on the same dye on different plants? This is a strategy that to increase the number of publication. If you are using the same thing, it is very difficult, because if you are staying in academic system, you need to have the publication also. Okay, that is the only reason that we are chosen like this. But our interest was to go with different plants which are responsible for that. The criteria, what we are chosen here, that the roots, growth of the plants, and mostly plants are the aquatic one. Okay, because we are dealing with the textile wastewater, so that should be the aquatic plants. Okay, that's why we are dealing with we have chosen the aquatic plants only, and this is the work done in our laboratory. And on this basis, the question comes that why you are doing this business? Because if you know that one plant is enough and plant can do that, why one should go for the number of plants? Answer is in the next slide. Next slide. Now, what we could find here, that the consortia systems put up a better fact. Only one plant cannot degrade the dye, but plant-plant consortium is better to work out. For example, here in first Rimazole orange R, time 36 hours, in consortium, you get the 100% degradation, but ester MLS, you get 42% degradation, and Glanduria pulchella, you get the 37% inhibition. Time is the same. It indicates these two plants combined together gives more degradation in less time. Okay, now it is very important. Time factor is very important. If you are saying that, okay, keep for 100 days and you will get the declaration or degradation, it is of no use, because we have to store that much water in a container, or you can say lakes here. You have to store that water for the treatment, and it is not advisable for the industry. That's why we made an attempt to use different plants in consortium and reduce the time. That is the only functioning, or that is the only criteria to how you can reduce the time of the declaration. Next, please. Now, if you can check here that again we tested here the enzyme activities, the same enzymes what we have seen earlier, and you'll find here in this increase in the or induction of the enzymes in one plant is different, and in another plant it is different. For example, lacase is induced, and lacase is present mostly in active form in Galladia pulchella, and it is induced 3 times, threefold. But we couldn't find that much lacase here. Again, that is a different type of the induction you will find here in different plants. That's why the rate of degradation is different in different plants. Two plants works in two different ways. Now, again, another observation what you can find here, if this is the individual plants with a particular dye. Now, if you are using these two plants in consortium, you will get different. For example, here you can say lignin peroxidase, which is increased here. Then lacase, it is increased in more form. Now here the increase here 0.09 is changed to 0.35. However, it is in plant alone 0.07 is changed to 0.1. See, that is the difference. That means that in presence of another plant, you will get more induction. In presence of another plant, you will get the more induction. That means the product of one plant can affect the enzyme activity of other plant. So two type of the effect you can get here. If you put two different plants together, two different type of the enzyme composition you are getting here, and as well the induction will be more. So metabolism will be the faster, and that is the reason that we got the less time. We could degrade 100% of that within less time. Next, please. That's why plant-plant consortium is better. Our laboratory was working for microorganisms too, and we have screened lots of bacteria, fungi, and the consortium too. So, and we could observe that the metabolism of dyes can take place by using the bacteria. With this values, we use another type of the consortium. Next, please. We tried. Yeah, we tried plant bacterium consortium. The plant is there, then you can have the bacteria, and the consortium is also there. And you will find here that at 56 hours, you will find there is no peak in presence of consortium. There is no dye present in the consortium. However, bacteria and the plants, there is a particular dye is present, but in consortium there is no dye. That means that the faster degradation can be observed in presence of plant and bacteria. Next, please. Okay, again, that is because of the enzyme levels present here. For example, in bacteria you will find isoreductase, but in plants you don't find isoreductase here. And induction pattern in case of plant is more lignin peroxidase. If you see 0 hours and 72 hours, in Ginea angustifolia is more compared to induction pattern in this bacteria 0.113 to 0.17. So various type of the composition of this composition of the enzymes is generated because of the consortium. So you will get the faster degradation. Next, please. Okay, these are the all chemical structures. You may not be see very carefully, but these the different only I can say that if you use the different plant or the bacteria and the consortium, you are going to get different type of the metabolites. And depending on the generated metabolites, that component will show the toxicity. Next, please. This is one of the experiment we did that what will happen if you take the contaminated soil and isolate the bacteria and induce that bacteria in the column, soil column, and check that what will happen. And we could, if you augment bacteria with the soil, you will get the faster degradation of the textile dyes. That means by bacterial augmentation will be better. It is naturally. And if you go with the KEZ pathway analysis, we'll find that number of the degradative enzymes are, we got the induction of these enzymes. Next, please. Okay. Now this, up to this, what we find that what should be the pattern? That the degradation of the dyes with the plants, degradation of dyes with the plants and plant consortium, degradation of dyes with the plants and bacterial consortium. Now, it is bioreactive, and in here, this is the textile effluent. We have developed some bioreactors. This is the textile effluent in the red container. These are the plant, Ganderia pulchata. This is the soil, and soil contains this bacteria. Now, for example, you can find here that the removal of TOC, removal of BOD, removal of COD. These are environmental parameters. If you talk about the removal of the mixture, yes, you can find that in control there is you can get bands, but in treated there are no bands. And if you see here in soil, the first line it is of soil, second line is of plants, third line is of bacteria, and fourth line, this is only 32 hours. This bacteria and the plant consortium degrade the textile dyes present in that effluent. That means this system is it works, and it is now, as earlier we have seen that bacteria can work, plants can work. Now here we can get the consortium of bacteria and the plant for the degradation of the textile dyes. Next, please. So you can see here that mostly we are interested in ADMI, that is the color is also decreased, and TDS is also decreased. Mostly I will concentrate on TDS because the phytoremediation system is important only because it reduces the TDS, which is not possible by the bacterial sources. Bacteria treatment of the textile effluent only by the bacteria will not reduce the TDS. Okay, that's why the phytoremediation is important for this textile dye degradation. Next, please. Now, again, earlier what we have seen that the system, and now it is a hydroponic system. We have used this Portulaca glandiflora, this in hydroponic way we have grown these plants, and we have circulated these textile wastewater. Again, you will find a tremendous decrease in the TDS. Simultaneously, COD, BOD is also decreased. Color is also decreased. Here we are checked with the textile effluent as well as textile dye. So if you can make a vertical system by using the hydroponics, yeah, this can be worked. It requires a less space. Earlier slide, we can show that the requirement of the space is more. Next, please. Okay, now this we have seen, now you can see the control, and we only in case of the bacteria, dye degradation from the soil. Then it is only plants, and this is the consortium plants and the soil. And if you find here, the consortium will be much more better to remove the residual dyes present in the soil. Okay, And if you see the plants, plant in third way is, the growth of the plant or the pigments are less here. However, the plants in the consortium are better. That means these microorganisms grown or supported microorganisms are helping to produce, grow the plants as well as degrading dyes. Okay, so here also residual also you can remove by the consortium of the plants. Next, please. Okay, now there are two systems available. One, that plants Ipomia aquatica, these plants are growing on the surface of the water. Just they can lie on the water. Certainly it is rooted somewhere in the soil, but just they can lie on the water and take part in the degradation process. In another system, you have some plants like Ipomia hederopolia. This requires a soil. This requires soil. So these plants we are grown on the soil. Okay. Now both plants in consortium, two different type of the characters we are used here. This is producing hydroponically, metabolizing the textile dye, and this bacteria and the plant system is taking care of the textile of the degradation of the dye. And that is coupled, we can say the coupled phytoreactor we develop for the which contains soil bed as well as rhizofiltration. Okay, so we have developed these two for the degradation of the textile dye. This is the laboratory experiments. Next, please. And now we were in the laboratory. And why we said the success story? That is because if you do work in the laboratory and show that, it's of no use unless you show in a real sense. And for that, we worked on HRTS 5-star MIDC Kagal, Kolhapur, and we developed the phytoreactor. This is the HRTS developed by the MIDC earlier, and this phytoreactor, this HRTS, we have converted into the phytoreactor. Okay, next, please. Okay, now this is our development, what we did. We are on the ridges, we have planted these plants, that is Patula, Aster then Portulaca glandiflora, and sorry, can you go earlier? And this is Gaillardia grandiflora. These are the plants we have grown on the ridges. Okay, there are two. If you find here, there are the ridges as well as the furrows. In furrows, you have the textile wastewater, which is generated by CETP plant, and in furrows we have planted here, and we are using this water to the growth of these plants. That means these plants can grow in the textile wastewater generated by CETP. Okay, so we have used this experiment for removal of the textile dyes. Next, please. Next slide. Now you can see here, Tagaceres pitipatula, this enzyme system we have checked, and we will find that most of the enzymes are getting induced in Tagaceres patula and grandiflora, Gaillardia grandiflora. These enzymes are, these two plants we found a better, have the better potential for the degradation of the textile dyes. Okay, next, please. Now you can see here how the degradation is taking place. We are adding the water, textile wastewater on the ridges. So it would have been if there is no plants, that will be continuous increase in the concentration of the textile dyes in the soil. When you use the plants, the concentration of the textile dyes is increased. That means we can remove the textile dyes from the soil by planting the plants on the soil. That is soil remediation also we can do it. Next, please. Now, as we have seen only the parameters, so number of parameters you can go with that. For example, pH, color removal, then metal ions concentration and all these things. Because here we are talking about the fertility of the soil also. If you use this textile wastewater for the degradation purpose, the soil fertility should not be decreased. And that's why we have to take care of that. And we could find here, yes, there is no fertility because the same ridge we are using at least for 3 times, because we work this project for 3 years. So 3 times we have used the base, but we couldn't find any decrease in the fertility of that soil. Next, please. Okay, this is another system what we have developed. This is the lagoons with the Salvinia mollusta. Textile effluent we are kept here, and these are the actual experiments on the site. And by using the mat of this Salvinia mollusta, we could degrade these textile dyes. And if you find here that all enzyme activities, particularly in the roots and the leaves, we are shown that mostly in the roots, you can find increase in the enzyme activities that directly indicate that they are working properly for the remediation of the textile dyes. Next, please. This is the system as we are progressing for the generation of the system. There are furrows as well as ridges also. We are used furrows for the water plants. We are used ridges for the soil rooted plants. And here there are 2 different plants we are used in the consortia, as earlier results showed that yes, consortia is better. So we are used that Vetiveria zizanoides and Ipomea aquatica. Vetiveria zizanoides, they grow on the, this is a rooted plants that requires some soil rooted plants. Ipomea aquatica, that's grown on the surface of the water. Combination of these 2 we are used, and we got the better conversion, better degradation of the textile dyes, by which we could decrease TDS, BOD, and COD. This you can see the first figures and the last figures. So this system also works in practice in the field. Next, please. Next slide. Okay, this is the phytoreactor we have developed in MIDC, Kagal, Maharashtra. This is actually is a 1 acre land. It is totally 45 acre land present there, out of which 1 acre land we have produced as a phytoreactor. And real results we are getting here, that there is a colorless water you can find if you throw this fully 1 acre, if you utilize this 1 acre land, you can get the colorless water. Next, please. Now this type of the furrows you can plant, and you can pass the textile wastewater from one side to other side. And if you make this type of the ridge, this type of the furrow till the river, so whatever the plant water is coming through, flowing through this furrow, that will be degraded. This is one of the possible way that to work out the real textile dye degradation, textile effluent degradation. Next, please. Now there is what we can find here. You can find here the roots: Alterantheria phylloxydroids, Cyprus flavidus, and Typhongus tifolia and Fabritus declama. Now you can see here, this is the laboratory one. Exactly this tank we have prepared, and we have shown that declaration here, and this type of the group development. Then we have made a tank, and we have shown that the floating bed by which we could get the declaration. And these floating beds we are used in the lake. It's a small lake over there, and this color water is also present there, and we can use this type of the floating beds for the removal of the textile dyes. Next, please. Now these are the results of laboratory experiment. You can see that the color is also decreased. Then BOD and COD and TDS is also decreased, because these are the most important parameters, and we could get the results by using Vetiveria zizanoides. Next, please. Okay, now this is our recent paper. We can make a floating bed park. So if you can, these are the all floating beds, different, And if you use, this is why we are using only one plant here, but you can make this a stream of water. You can make a number of floating beds like this. It is taking care of degradation of the textile dyes, flowing this water through nalas from textile industry side to the river. Now you can imagine this is the river to that side and the development of this. And if you can prepare this type of the floating wet park, and this floating wet park can be used for the removal of the textile dyes. Actually, this idea, or you can, if you can prepare the microcosm, and in that microcosm you can make a floating wet park, and that will take care of the degradation of the textile dyes. Next, please. Now, mostly that everybody bothers because, this is the removal of the textile dyes and nobody wants to spend money on that. That is a major problem here. And that is why we get the pollution in India. Okay. And nobody bothers also. They establish only CETP plants and just they bother that is working or not. Okay. And there are different ways that they can do that things. Okay. Now, how to reduce the cost of that thing? How to reduce the cost of phytoremediation? Certainly, it's a low-cost system, but again, if you reduce the cost of phytoremediation, so we tried that. Can we develop the electricity? We have used the microbial fuel cells, and if you use the microbial fuel cells, this is you can see the system electrified floating phytobed technology for wastewater treatment. If you produce this type of the system, that will take care of the generation of the electricity as well as degradation of the dyes. And what our observation is that if you make this type of system, dye degradation will be the faster. That is one effect we are getting, and there is a generation of the electricity. But only we could produce a very small amount of the electricity produced. Certainly, this technology may not be advisable, but if you reduce the cost of cathode and anode, and if you can make the system more appealable, yes, this system can be used, by which you can reduce the cost of the system also. Okay, so I think this is also recently we work on this. Next, please. Okay, so here in the last slide, you are finding here that the water is flowing through the nalas, as I mentioned. These are the plants on the soil. They are build up the soil. They are phytoremediators. These are the they are also taking part for the remediation as well. There are some floating plants on the water. They are also taking care of this. If you create this type of the nalas to reach textile wastewater, treated CETP treated textile wastewater to the rivers, yeah, this system also can be used to decolorize or the degrade textile wastewater, and clear water you will get in the rivers. Okay, thank you very much for your patient listening. If you have any questions, I'm happy to answer.
Thank you, sir.
Thank you, sir. It was indeed an insightfully inspiring session. This is truly a story of how small in vitro health experiments could ultimately lead to large-scale applications and could find a long-term as well as a sustainable solution to serious pollution issue. It's really a fascinating lab-to-land approach. Thank you again for the insight, sir. With this, we would like to open the question and answer session, and I would like to request the audience to kindly drop in your questions. We actually have a few questions here. First question is from Harshil Deshmukh. Can all plants and bacteria be used in a consortium, or is there a specific compatibility criteria so that maximum degradation is achieved?
Yeah, there is certainly compatible characteristics are there. Now, if you use some plant system, very few type of the bacteria they are growing in the rhizosphere of that plant. We have to take care of that, what type of the bacteria they are going through the rhizosphere. But mostly if you are using the textile dyes for a longer time, the bacteria which are present there, that will take care of the degradation. certainly. Not
True, sir.
necessary to bring the bacteria from some other places. That will take care of that.
He has another question where he's asking, when should the plants deployed for phytoremediation be harvested?
Yeah, that is it's a good question. Yeah, plants can be harvested. You have to cut, as I mentioned here. Mm, in some of the slides, there is a harvested plants also. You might have seen that. I have shown the flowers, the flower
Yes,
beds.
sir.
There were eucalyptus plant, and they have harvested that. And mostly in HRTS, you can find the eucalyptus plants. It is for transpiration of the water, not for the degradation of the textile dye. It is for the transpiration of HRTS. Main function is the transpiration, not the degradation of the textile dyes.
So the next question is from Govind Vyavahare. Is there any chance to get genetic mutation in the plants which are being used or being employed in phytoremediation systems?
I don't think so, because yeah, these are the chemicals what we are dealing with. The mutagenic and the carcinogenic chemicals are there, but because the plant life is not long life, it is only for a particular cycle, 6 months or 1 year. So I don't find anything here. But whatever plants were being used is yes, they were the good growing plants.
Okay,
But
sir.
I can't deny the possibility. I can't deny.
So the next question is, please continue, sir.
I can't deny the possibility because we are working on the carcinogens and mutagens. The possibility is there, but I don't think that is last long for number of times. It is, there was a question of the transgenic plants. Nobody earlier, India also not allowed to use transgenic cotton. Only was this type of the bothering people say that okay, that gene might hurt something else. But nowadays we are using transgenic plants. Nothing is going to happen. That type of the question you can get. Okay, that is the importance to be given only to that point.
Yes, I think that really well answered his question. The next question is by Yash Patil. Does it need stagnant water necessarily?
Not necessary, because in another experiment, floating water we are used in one of the experiments, hydroponics. That is the floating water.
Okay. Then there's next question by Dr. Smita Shinde. Eucalyptus trees are banned nowadays by Forest Department. So what are your comments on that?
Yeah, eucalyptus trees may be banned in, may be banned somewhere where the water scarcity is there. Eucalyptus tree is mostly used for the transpiration of water. Here we have to suck the water and transport in the air. We are using for different purpose,
Yes, sir.
And we have to have some excuses all the time. Mostly, we have some, for example, it's a very basic question. I will extend my answer. One question has been asked to me when I was presenting my data to the Department of Biotechnology. And I have shown that the effluent has 4 gram of TDS. And 4 gram TDS, 4000 milligram TDS, that water cannot be used in the soil. And that question, they have asked me that, okay, professor, that your effluent contains 4 gram of TDS and how you can use in the soil. But the question is that the plants, in special cases, the plants are growing in that TDS. And they are taking care of the degradation that degrades to near about 1 gram, which is advisable for the watering the plants. So that type of the excuses you have to make it. There is no alternative for that.
Okay, sir. So we have another question. How is this technology and its application scalable in different geographical locations around the world?
Only that we have to find out the different type of the plants, depending on the geography. The same system, whatever the plants we have done in Kolhapur may not be available in USA or the Korea. So we have to choose the different type of the plants. And we have to say whole thing what it did in last 10 years, we have to do in the same place, and we have to find out the bacteria as well as the plants in different ways. What are the different aquatic plants are available? But mostly typha is all over. You can use the typha. Then you can use aquatic also. I use aquatica and typha in South Korea too, and in India too. They are working, but we have to find out that we have to take care of the growth of the plant. The plant should be growing there.
Okay, sir. So, is there any specific category of dyes which are really complex or which are really difficult to be degraded by the plant microbial consortium?
As far as our study is concerned, that yeah, all dyes are getting degraded. They absolutely, there is no problem. We say that dyes are difficult to degrade in the sense that in natural environment. We are inducing every system here. We are talking about the enzymes now. We are inducing the enzymes. We are gathering, augmenting this particular sources with different type of the good bacteria, those who are taken care of. So there is no problem. This can be degraded. There should not be any problem.
So we have another last question. Can we use cloned bacteria for phytoremediation?
Yeah, that is, but yeah, answer is, I can say, theoretical answer is yes, we can use, but if it works. Clone bacteria doesn't work for a longer time. Only one thing is very important here. We are not using it, we are not working in the laboratory. Whenever you find the clone bacteria, transgenic bacteria, whatever it may be, that is in the laboratory in a very controlled condition. We are working in uncontrolled condition. So there is a very less possibility to work with the bacteria, that engineer bacteria with this. Yeah, but if you go, we can make a transgenic plants, because this was my aim to do with transgenic plants. That mostly very good observation what I find here, that the plant lackages, although plant lackages are more in quantity, they are not potentially degraded of the textile dye, but bacterial lackages are there. So if you clone the bacterial lackages in the plant cell, yeah, certainly that will be the powerful system. But since long I'm trying, nobody is ready to work in plant system with the bacterial lackages. I do not know why. Because I don't want to clone that plant in standard plant. Okay, I want to clone in, for example, Tagatus patula, and Apomia aquatica. People, those who are working plant tissue, they bother about the aquatic plants. They don't go for the plant tissue culture. So there may be some interested fellow which is working in the plant tissue culturing the aquatica, Hypomia aquatica, and the cloning of the bacteria. There, that will be helpful, certainly. That is what we can say, the transgenic plants, cotton plants work for the control of the pest. Similarly, yes, that can be done, but it is very difficult.
Absolutely, sir. Thank you so much for answering our questions.
Marzuka, 1 minute. Sir, one last question from my end.
Okay.
So is there any initiative taken by government of Maharashtra or government of India or Ministry of Environmental Science to develop this technology at different industrial level or different textile industry, MIDC area? Because I have seen we had got a number of projects funded by DBT and DST. Then Ganga River cleanup project was also there. So is there any technology development by government?
Yeah, sir, I'm happy to share here. I was a task force member in DBT, and we have completed one project and give the answer also. And we have worked on, when I was in Korea, one of my colleague, your guide, Professor Jyoti Jadhav, was working on that project. And it's a multi-institutional project, And successfully we have completed that project too. And that time, Department of Biotechnology, particularly environmental, they were interested to start up with different locations. But nobody came forward to do that because it is a complicated story. Simple reasons that what we do in India, we have the technology, but we are not using the technology properly. And nobody wants to do that. Because if I do the same work, if there is no objection from anybody, if I spend 1,000 rupees, why I should spend 10,000 rupees for that? That's the question. So that's why nobody is coming up for the cleaning of the textile dyes. That's why the problems are there. If you check every, I think in rainy season, Teachal Karanji people starts havoc. Why? Why there is no constant solution there? All technologies are available, but nobody is using. That is the problem in India. And another problem is also there. For example, in my case also, I'm coming from a very small university, and I started, and at the end I went to the DBT as a task force member. And after 1 year, I retired from India. So it was not just possible for me to extend. This type of the things are happening all the time. Nobody wants to extend, and mostly nobody wants to work in the field also. Because as you will be aware of that, in the field you would get the publications. You can get more publication using the bacteria and working in the laboratory. These are also negative points there. But certainly I will be happy if somebody is elaborating this system in the field. I'll be happy.
Okay, sir. Thank you. Thank you.
Thank you, sir. The session was quite interesting and hopefully helpful as well as inspiring to many of the students. I would like to request our guest speaker, Professor Govindwa, sir, to give us concluding remarks before we wrap the session.
Do you want me to talk
Yes,
something?
sir.
What I should talk? I talked about 1 hour. Okay. As I say that you explained that what is the phytoremediation, but this is a story that actually it is a build-up story, and it is a real story. Because we started with one dye and which one bacteria, and mostly, in 2006 we have published one paper, degradation of textile dye by some, and we were only looking for the pH, temperature, this type of the parameters. And we developed in such a way that we could develop the different type of the bioreactors, and we could implement this to the field. Actually, this MIDC area, we have about 1 acre land there available. It is at today also it is working. And interest part is all these DBT members. They came to visit the site. Really, this happens in India or not? That is, I appreciate their efforts. That okay, they come here, and this is, I am feeling proud that at least they visited my place to see that the system works. And anybody, those who are working on the phytoremediation, I see they can proceed further, extend this research further. That will be I'll be very happy.
Thank you, sir. I would now like to request Dr. Rahul Khandare, the co-convener for the session and Associate Professor of Amity Institute of Biotechnology, to deliver the vote of thanks. Over to you, sir.
Yeah, thank you, Marzuka. Let me take this opportunity to personally thank you, sir. Sir is my mentor, my guru. He is architect behind this entire design, entire research work. We could contribute a little under his guidance. Thank you very much, sir. On behalf of Amity University, Mumbai, Amity Institute of Biotechnology, I'm very much thankful to you for an insightful deliberation of yours. Thank you very much. I'm also thankful to Honorable Founder President, sir, for being a source of inspiration. I'm thankful to Chancellor, sir, for motivation and guidance. I'm again, my thank go to our Vice Chancellor, sir, for all the support our Pro Vice Chancellor Sir for all the permissions and active participation in such events. Most importantly, the head of the institutes, head of the departments who have joined this event, and our director who has been constantly guiding us to have such insightful and thought-provoking sessions, lectures for our students. And last but not the least, all the faculty members of AIB and the university, and also the most important part of such events are bright students. I thank you all. Thank you, and thank you again.
Thank you.